Thank you so much. I'm Rossella Breveglieri from the University of Bologna. And today I will talk about the spatial attention and posterior parietal cortex. But first of all, I would like to thank the Brainbox company for two reasons. One is because they helped me a lot in setting my new TMS lab some years ago. And the second reason is this invitation, because for me it's a very nice opportunity to present the data of my previous lab of electrophysiology with monkeys when I was educated in research, and also my new data of my new TMS lab, and of course also data of other labs. So we can start by saying that attention is a very general word, because we can have different types of attention. So we can orient attention to different spatial locations. So we are talking about spatial attention. But we can also focus our attention to specific features of, for example, visual stimuli. And so in this case, we are talking about feature-based attention. And there is also this general alertness, which is a kind of mental state with that we can use, that we have to use in our everyday life when, for example, we are driving. And in this talk, we will focus on spatial attention, but we will also have some data about alertness. When we talk about orienting spatial attention, we can have exogenous orienting of attention and endogenous orienting of attention. When we talk about exogenous orienting of attention, we talk about an automatic procedure, a kind of external orienting, a bottom-up. Is when we automatically orient attention to a sensory stimulation. On the other side, when we talk about endogenous attention, this is a kind of a goal-directed process, a kind of a top-down. It is called also internal orienting of attention, and this happens when we voluntarily move our spotlight of attention in one spatial location, for example. When we talk about orienting of spatial attention, we can also have covert orienting of attention or overt orienting of attention. So let's make this example. If this guy is looking at this girl and he is also paying attention to this girl, we are talking about overt orienting of attention. Whereas if this guy is looking at this girl, but simultaneously is paying attention in another spatial location without moving the eyes, this is a covert orienting of attention. And in this slide, we will focus on covert orienting of attention. So spatial attention is very important because it is needed to sample space, but also to improve perception and to interact with objects. For example, if you want to hit this ball, we have to pay attention there. So without attention, many manual interactions would fail. And different brain regions are involved in maintaining spatial attention in one position or shifting it to different spatial positions. And one of this region is the posterior parietal cortex. So in this talk, we will focus on the medial part of the posterior parietal cortex. And here in the macaque brain, we can see a posteromesial view of the right hemisphere of the macaque. And here you can see I want to see if I can use the laser. So here you can see the parieto-occipital sulcus, and in color you can see area V6A, which is in the anterior band of the parieto-occipital sulcus. And this is a part of the medial posterior parietal cortex, and this is the area of interest of this talk. So in 2010, we have recorded single cell activity of area V6A neurons in the monkey, and the monkeys were trained to stay in front of a screen, and they were pushing a button close to their chest, and after a while there was a fixation point. So the monkeys had always to fixate in this central part of the screen. So after a fixation period, there was a cue briefly appearing. It was an exogenous cue, a peripheral cue. And after the cue offset, there was this delay, and after this delay, there was a target to detect. So they had to pay attention, covert attention, in this previously cued location, because there was this target to detect by a manual response. They had to release this button. And during this task, we recorded the single cell activity of area V6A, and this is the population discharge expressed as a spike density function of cells modulated by this task. In particular, this is the cue onset, so this is the delay. So in this epoch, we had this shift of the spotlight of attention to the cued location, and we can see here that there is an activation of the population of cells for this shift of the spotlight of attention. It was about 30% of the recorded cells. And so this was one of the first proof of concept that V6A neurons fire during covert shifts of the spotlight of attention. After a while, in 2015, there was a nice parallel of this data in an fMRI study with monkeys performed by the Wim van Duffel's lab in Leuven. And what they did is to train monkeys to fixate on a screen in the central position. And there were two visual stimuli, visual shapes, and the monkey were trained in paying attention only to the relevant stimuli, because there were couples of stimuli, but only one was relevant. So in this case, the relevant was a circle. And they had to ignore also the irrelevant stimuli. Okay, so in this case, the relevant stimulus was to the left, and they had to pay attention there. And after a while in the trial, these two stimuli disappeared, and the other two stimuli appeared on the screen, and now the relevant stimuli was to the right. So they had to shift the spotlight of attention from the left to the right part of the screen. They had to pay attention there because they had to respond they had to give a manual response to the dimming, but only of the relevant stimuli, while ignoring the irrelevant ones. So in this trial, we had a shift of the spotlight of attention, of the covert spotlight of attention of the monkey. In other trials, there was a relevant stimulus here, and then another relevant stimulus, but always in the same spatial location. So in this trial, they have the maintenance of the spotlight of attention. So the spotlight of attention stayed there, was not shifted. And the monkeys performed these trials, this task in the scanner for fMRI. And what they found is summarized in this it was a flat map of this hemisphere of the monkey brain, and area V6A is here, as you can see. And if you see colors in this map, this means that there was a significant activation resulting from this contrast, shift of the spotlight of attention versus stay. So as V6A is colored, this means that V6A is activated during covert shifts of the spotlight of attention. This is the 2D map resulting from the contrast stay left versus stay right. And you can see here that V6A is not activated. So V6A is not activated during maintenance of spotlight of attention. More recently, a human homologue, a putative human homologue of the monkey V6A was found in the human brain by fMRI studies. So you can see here the same view, a posterior mesial view of the right hemisphere of the macaque and of the human brain. And in red, you can see the position of V6A in the monkey and in the human brain. And V6A is always in the medial part of the posterior parietal cortex, but with a very important difference, because in the human brain, V6A is superficial. So this is a very important point for us, because this means that in the human brain, we can use transcranial magnetic stimulation to study the causal role of human V6A. And one of the first TMS studies over human V6A was performed by Chavarro and collaborators. They instructed participants to stay in front of this screen, and there was a fixation point in the center. So here we have covert attention again. After the fixation time, there was a cue, an exogenous cue, peripheral cue, appearing on one of the two positions, and then there was a delay, and they stimulated the homologue of the human V6A here with the repetitive TMS online. And after this delay, there was a target to detect. In the majority of trials, the target appeared in the previously cued location, and these are called valid trials if we want to use the Posner nomenclature. And in a minority of trials, the target appeared in the opposite location. So these are invalid trials in Posner nomenclatures. So in valid trials, the participants maintained attention in the previously cued location. In invalid trials, participants shifted the attention in the opposite location. And what they did is to record the reaction times for target detection in valid trials, the white data, and invalid trials, the black data here. And this is the distribution in different stimulation conditions. So the sham condition, of course, is baseline with, let's say, no stimulation. And we have the stimulation of left V6A and right V6A. And what they found is an increase of the reaction time, which means a kind of impairment in the target detection after stimulation of V6A compared to the sham condition. But they only found this in invalid trials, so when there was a shift of the spotlight of attention, and they didn't find any TMS effect in valid trials. So they suggest that V6A in the human brain is involved in the redirection of attention and not in the maintenance of attention. So to recap, monkey and human studies suggest that the medial posterior parietal cortex is involved in the shifts of spatial attention and not in the maintenance of attention. And this was the result of attentional tasks. There is another bunch of study. This is just an example. This is just an example. In the macaque with single cell recordings, we have recorded cells activity in V6A when the monkey was performing a reaching task. So this is a peristimulus time histogram, which is the representation of the response of one single cell. When the monkey was planning a reach and executing a reaching movements towards different spatial locations. And we can see here that there is a peak in the histogram, which means an activation of the cell during planning and during execution of reaching. And we have a nice parallel here in a human TMS study performed by Michael Vesia and collaborators. And they have instructed participants to fixate a central position on a screen. Then there was an instruction. Here we have an R, because in this study they also had saccade trials. And then there was a cue appearing, a peripheral cue, and then a mask, and then there was a delay. And in this delay, they had to plan a reaching movements towards the previously cued location, and then they had to reach on that location. And during the delay, during reach planning, they delivered TMS over human V6A. And here we can see the results of one representative participant. You can see here that you have ellipses. Ellipses are the representation of the precision of reaching. In red, we have the precision of reaching to the left or to the right, in trials where they stimulated V6A. And in gray, we have ellipses, so the precision of reaching during sham stimulation. And you can see here that here you have a difference in precision. So a big ellipse means that the reaching is less precise. So they suggest that TMS during reaching preparation on V6A, so during reach planning, resulted in a less precise reaching. So to recap again, monkey and human studies suggest that the medial posterior parietal cortex is involved also in reach planning and execution, and this is the result of a reaching task. So shift of spatial attention and reach planning and execution. However, usually planning of reach requires attention to the reach target. So if we want to hit the ball, we have also to pay attention there, as I've also mentioned before, unless we are forced to behave differently. So we can ask, can the role of human V6A, so the activations that have been seen during reach planning, be explained only by the allocation of attention? So in other words, is human V6A involved in reaching per se? So to answer this question, we have to design an experiment where reach can be planned towards attended or towards unattended targets. And we did this, and we have designed a transcranial magnetic stimulation experiment. But first of all, we have to say that manipulating attention and reach planning independently is very, very difficult. But there was one study in 2021, a monkey study by the lab of Stephen Wise and Aldo Genovesio. This is a monkey study, so a single cell recording study. And they have designed a task where they separate attention from reach planning. Basically, the monkey was in front of the screen, it was fixating in the center, and then there were targets appearing on the screen, and then there was a cue. So this cue was double informative because the peripheral part of the cue had a color that was informative about the location to plan subsequently a saccade. So if the peripheral part was red, this means that the monkey has to plan a saccade to the right of the screen. The central part of the cue was informative about where to pay attention. So the blue color means that they had to pay attention in the upper part of the screen because they had to detect the brightening of this particular target, the upper one, and this was the go signal to perform the saccade. They have different combinations of colors. So in these, when the colors of the peripheral part of the central part were the same, this means that attention and saccade planning were on the same location. And with different colors, this means that the attention and saccade planning were on different locations. So it was covert attention. And this is the distribution of the reaction times of the monkey to the saccade initiation. And what we can see here is that we have two colors, two graphs. The blue one is the one where attention and saccade plan were on the same location, and the red one, different locations. And we can see here that for both monkeys, the reaction time for conditions where attention and saccade plan were on the same location, were congruent, reaction time were faster, as expected, but this was the proof of concept that the task was effective. And basically what they found is that they found different populations of cells in the frontal cortex, in prefrontal and premotor cortex. There was a population modulated by motor planning, another population modulated by attention, and there was also a very small population modulated by both processes. And so we were inspired by this task, and we slightly changed this for human participants. So we have instructed participants to stay in front of a screen. There were two targets, and then there was a fixation point. The right hand of the participants was close to the chest. They had to press a button, and after a fixation period, there was a cue. And the cue was double informative because the color of the cue was informative about where to plan the reach. Here we have a reach, not a saccade like in the WISE lab. And so red means in this example that they have to plan a reach to the right, a subsequent reach to the right. And the colored side was informative about where to pay attention. In this example, they had to pay attention to the right because the colored part was the right one. And during this delay, so when motor plan and attention were allocated, we had stimulated with TMS human V6A to understand the role of V6A in this interplay between motor planning and attention. So after a while, there was the target to detect in the cued position, and the target was the go signal, because after the reaction time, the participants had to reach that position, the position that was instructed by the color of the cue. So we had different types of trials. The trials were motor planning, reach planning, and attention were in the same spatial locations. Here we call them congruent trials. So in these trials, reaching was performed toward attended locations. And we had also different kinds of trials where attention was here, but motor planning was there, or vice versa. But in this case, reaching was planned towards an unattended location, so incongruent trials. We had also a kind of control trials where the cue was only informative about the location of motor plan, and attention was not constrained because the cue was fully colored. And so we have also these unconstrained trials. So we have collected the data from a group of participants, healthy participants, and this is the distribution of the reaction times in the different types of trials. We have congruent trials, so reaching to attended targets, and also incongruent trials, reach and attention in two different locations, and unconstrained trials. These are the data during sham conditions. And we can see here that reaction times of congruent trials, so when reach was planned towards attended targets, was the faster. So significant differences, and as expected, but this again was a proof of concept that this task was effective in manipulating attention and reach planning. But what happens to reaction times in transcranial magnetic stimulation trials? Our hypotheses were that if V6A is involved in reaching per se, we should see an effect of TMS in all the trials. So when reaching is planned towards attended and unattended targets. On the other hand, if V6A is not involved in reaching per se, but something more complex is going on, TMS effect should be seen only in some conditions. So what we have found? We have found the results of TMS only in congruent trials. So only when reach was planned towards attended targets, we saw an increased reaction time, so a kind of impairment. And so TMS over human V6A slows down the reaction time only when reaching to attended targets. So V6A seems involved in reaching, but only to attended targets. We didn't find any TMS effects in other trials. We have also recorded the pupillary response as an index of arousal, and we tried to correlate this pupillary response to cue appearance, so during the delay, with the reaction time. And we have found in sham stimulation, but only in congruent trials, so when reaching was planned towards attended locations, that the pupil response was predictive of the reaction time. And what happens during TMS? During TMS, after TMS over human V6A, this correlation was impaired. So we can suggest that V6A in the human brain is involved in the coordination, arousal, reaching, but only when planning a reach to attended target. So remember our hypothesis, V6A is involved in reaching per se. We can discard this because we have found TMS effects only in reaching to attended targets. So we can confirm the second hypothesis. So V6A is not involved in reaching per se. So let me conclude this talk by saying that V6A in the human brain is not involved in the maintenance of spatial attention per se, tested during attentional tasks, as we can see in the literature. Our data suggests that V6A is not involved in reaching per se, but in reaching to attended targets. So putative V6A, the take-home message, is involved in attention for reaching. So now let me thank all our collaborators, our colleagues at the Department of Biomedical Neuromotor Science here at the University of Bologna, at the University of Münster, and in the Department of Psychology here at the University of Bologna, and all of you for your attention.